Today, Caroline Slomp will introduce the topic, and afterwards 2 speakers will tell something about their research. Caroline Slomp is from the University of Utrecht in the Netherlands, and Take Dalsgaard, if I pronounce it right, is from the Aarhus University in Denmark, and Lisa Levin is from the Scripps Institution of Oceanography in California. So, Caroline, I would like you to ask first.
Okay, thank you very much. I'll be showing some results or discussing some results about oxygen depletion in the sea, as will be presented tomorrow on Thursday in a session on low oxygen. Why is this so important? Well, oxygen is essential for fish and fauna living in the sea, but there are more and more places where oxygen is low or absent in seawater. And so what we see is that so-called dead zones develop. And so you can actually see this here on the slides. You see the right-hand side, you see zones of the seafloor where animals can no longer live and where bacterial mass are present, and higher life forms are they're limited. We call this triple trouble because actually this low oxygen or part of it is a consequence of the increases in carbon dioxide in the atmosphere. And we all know that that's leading to climate change and to ocean acidification. But low oxygen is a third major problem that is linked to global warming. So how does that actually work? Well, we have areas of low oxygen that naturally occur in the ocean. So we have these, for example, in the Pacific. So they mainly occur along the western side of continents, so in the Pacific Ocean, along the coast of Namibia, and also in the Arabian Sea. But what we are seeing today is that these areas are of oxygen minima, we call these oxygen minimum zones, that these are expanding. And this expansion is due to the fact that due to global warming, the ventilation of the ocean's interior is changing. So the intermediate and deeper waters are not so well oxygenated, simply because warmer water will not as easily mix and will just stay on top of colder water. And there's another effect, and that is that oxygen solubility is reduced with rising temperatures. And so what we see is that in the open ocean, these zones are expanding. And so what Take Dalsgaard and Lisa Levin will discuss is some findings, new findings about these open oxygen minimum zones that will also be presented tomorrow. And what Tatega finds, for example, along with co-workers along the western coast of South America, is that there's a layer of oxygen-depleted water that with concentrations of oxygen lower than known before, and that actually that's a very important zone, and that it actually impacts nutrient cycling in the ocean. And Lisa Levin will show that human activities are actually affecting these oxygen minimum zones. And even though we don't know that much about the processes occurring there, humans are actually mining there and are removing oil and doing all sorts of things. And Lisa will also show what kind of animals live there, and also how they colonize, or how their colonization is affected by low oxygen. Now, it's important to realize that low oxygen not only occurs in open ocean systems, but also in coastal systems, so in our coastal waters. And this is a map that was published 3 years ago, but shows that in 400 coastal systems at that time, these so-called dead zones had evolved. And in this case, it's not only global warming, but a major factor is, in fact, the fact that nutrients from land are being brought into the sea. So nutrients, so nitrogen, phosphorus, are coming in from fertilizer and wastewater into coastal waters, and they're fueling growth of algae. And when these algae die, they need oxygen to degrade, and that creates an oxygen demand in the bottom water. And so the oxygen can be depleted. And one of the key zones where this is occurring is the Baltic Sea. And actually, this is what is often called the largest dead zone that's caused by humans in the world. And you can see on this map, you can see the maximum extent, the area of hypoxia in the Baltic Sea, this red area. You can see it's a very large area. So, well, what is the cause? As mentioned already, nutrients from land to a basin that is prone to poor mixing can create this problem. And if you want to do something about it, you need to reduce nutrient sources. So if you want to reduce this coastal hypoxia, so you need to do something about the fertilizer and about the wastewater that's leaking into these systems. And in some cases, that's being done. And a very interesting result there is that in one of the coastal regions in the Baltic Sea, in fact, where wastewater has been removed, in fact, something interesting has happened, and that is that a worm that came into the system here is actually helping ecosystem recovery. So wastewater reduction has actually led to a condition where there's a little bit of oxygen, and this worm, this invasive worm species, has come into the Baltic Sea, so it didn't exist there to start with, and it has started to irrigate the sediment, so it's pumped oxygen into the sediment. And this has led to chemical processes leading to removal of nutrients. And what you can see in this figure is actually the more of these worms you get, the more retention of the nutrient phosphorus in the sediment until a certain maximum. And so this is actually a nice positive result where an invasive species, so a new species in this system, is actually having a positive effect. And with that, I'd like to give the word to Tatega, who will continue about the oxygen minimum zones.
Thank you. Just need to get it switched. I'm here. Okay, I will present some results from the about oxygen and nitrogen in the South Pacific oxygen minimum zone of Chile and Peru. I show a map a bit similar to what Caroline just showed, showing the oxygen minima in the oceans. And these, the more dark red and black it gets here, the lower are the oxygen concentrations. There are these three major oxygen minimum zones of the ocean. There's the Arabian Sea, there's the eastern tropical North Pacific, and the eastern tropical South Pacific. And this is down here where we have worked, so that's where I'll concentrate on. Down here, the surface waters contain oxygen. When we get down to about 50 meters or so, oxygen gets depleted, so there's very low or no oxygen, and then this continues down to about another 300 to 500 meters down, then oxygen appears again. And inside this oxygen minimum, there's a lot of nitrogen cycling going on. I want to show some main points, first regarding oxygen. The reason we started, or one of the things we did there, was that colleague Niels Peter Revsbeck developed a new oxygen sensor, which was about at least 100 times more sensitive than the previous methods. That means that we can now detect oxygen down to 0.01 micromoles per liter, and previously the methods went only down to 1 to 2 micromoles. So that makes a big difference here. The reason that we are interested in the oxygen concentrations here is that when oxygen gets very low, microorganisms stop using oxygen and switch to other life forms, one of which is removal of nitrogen from the sea. And talking about nitrogen, the oxygen minimum zones are very important there, because these three major oxygen minimum zones, the Arabian Sea, the northern, the southern, eastern tropical Pacific, they remove 1/4 to half of the global marine nitrogen removal. So they play a large role in the nutrient cycling of the oceans, even though they don't cover very large areas. Nitrogen is important because the availability of nitrogen regulates the growth of algae in the sea. And so if there's more nitrogen, then there will be more algae. And the algal growth removes the carbon dioxide from the atmosphere and therefore counteracts the rise of CO2 in the atmosphere. And what we've done now is looked at the oxygen concentration, or the threshold concentration, below which the microorganisms switch from oxygen respiration, oxygen breathing, to the other life forms they can do, and remove nitrogen. So just talking about oxygen, there's different scales. So this is a scale of oxygen where we look at the surface waters in the sea. There are about 300 micromolar of oxygen. If we go down to half of that, the fish will start to escape, try to avoid it, those that can. And if we go another 50% down, the fish and macro life will start to die. If we expand this scale now by a factor of 100, we can see now the old detection limit of the known oxygen methods. And if we expand this even further by a factor of 50 here, we can now see the new detection limit. This is very low here. And the reason this is interesting is that microorganisms are very, very good at taking up oxygen from the environment. So if there's more oxygen than shown here, then most microorganisms apparently see the environment as fully oxygenated, so they can keep growing and living as if there was full saturation of oxygen. First, when they get below this level here, they start to become limited by oxygen. They simply slow down and start shifting into other forms of life where they can remove nitrogen. And that is why it's important to be able to measure nitrogen in the same range as the bacteria see the nitrogen. Measure oxygen in the same range as the microorganisms see the oxygen. And nitrogen, as I mentioned, is very important. We have a lot of nitrogen on this planet. A lot of it is in the atmosphere. This is symbolized by the green circle here. 80% of the atmosphere is nitrogen, but this is not directly available to plant growth and to algal growth. The little red dot down here symbolizes the amount that's available for algal and plant growth. And the size of this little red dot is the one that determines the growth of algae in the sea. The size of this one is regulated by the balance between these two arrows. The green one representing the processes that can transform atmospheric nitrogen into available nitrogen. The blue one represents the processes that do the opposite, take the available nitrogen, remove it, and transform it into atmospheric nitrogen. We did a cruise in 2007 along the coast of Chile and Peru, where we measured the nitrogen removal experimentally on these stations indicated by the blue letters here. And then we also measured oxygen on a number of stations. And this thick red line represents the cruise track where oxygen was below the detection limit of our new methods. So here, on this 2,500 kilometer long cruise track, oxygen was so low we couldn't see it with this method. And this has implications for the nitrogen cycle and nitrogen removal. And these three processes mentioned here are the ones we know at the moment that remove nitrogen. You don't need to know the names of them, but the common thing for all of these three is that we only measure high rates of nitrogen removal by each of these processes if oxygen concentration is below detection limit of our new method. Only in a few instances here and here and here and here we see a little nitrogen removal even in the presence of oxygen. But otherwise, oxygen apparently needs to be below the detection limit to occur. So the conclusion is that we can now measure oxygen at much lower concentrations. We find very, very low concentrations in the oxygen minimum zones, and we need these low concentrations apparently for the nitrogen removal to occur. And with this, I would like to stop here. Yeah. Okay. Do you want this one?
Can you help me pull this up on the PC? Yeah. Where is it? Where is the cursor? Okay.
I can do it from there, from the beginning.
So, yeah.
And so to advance it, where? Okay.
Or the mouse.
You're going to click the mouse. Yeah.
Okay, we'll try this. Okay. Well, as you've heard, the world has some major low oxygen zones termed oxygen minimum zones, and these are midwater features shown here in purple throughout much of the eastern Pacific, West Africa, and in the northern Indian Ocean. And where these intercept the continental margin, they create hypoxic or low oxygen seafloor. We've estimated that over 1.1 million square kilometers of continental margin is in fact hypoxic or experiences extremely low oxygen. Now, these same continental margins, in addition to the stress of low oxygen, experience tremendous pressure from human activities that are moving into deeper and deeper water. These include fishing, which has gone from the shelf now to the continental slope. Our oil and gas extraction activities have gone deeper into some of the margin waters, also experiencing low oxygen. And in some countries, such as Namibia, we're now mining minerals like diamonds and phosphate, which is a product of this very low oxygen. Mining is just about to begin over some big swaths of the oxygen minimum zone in Namibia. Now, as you heard also from Carolyn, the global warming is causing reduced ventilation of the ocean waters and thus declines in oxygen and increases in what we call hypoxia. And this means also that the oxygen minimum zones are expanding and shoaling. And this is a process that we call deoxygenation of the oceans. And as she mentioned, it's a relatively little known side effect of CO2 addition into the atmosphere. And I'll show you some experimental results now that help us understand that deoxygenation in the ocean is likely to reduce ecosystem resilience. And I guess what I forgot to mention on the previous slide is that understanding human impacts and proper stewardship of our continental margins require that we know something about the resilience and response of these ecosystems to disturbance. So to try to understand this, we conducted a series of experiments, basically colonization experiments, in which we looked at the short-term recovery of animals on the Indian margin. And we did these experiments at 3 locations here. This is the oxygen profile across the Indian margin off Goa, on the western side of India. And we put out a series of colonization trays, broad trays filled with defaunated or azoic sediments with no animals. And we set them out 4 at a time, and then sampled after 4 to 8 days to look at which animals had recovered in these sediments. We also sampled the background sediments or the ambient sediments surrounding these to see what the source populations would be. And these experiments were done with the Shinkai 6500 on a Japanese research vessel. And here I show you just very briefly the results. We had no colonizers coming in. Can I go backwards? This, I think what I needed to say that's quite important is we put these out at 3 different water depths. At 540 meters, we had extraordinarily low oxygen measured at 0.1 micromolar. And then at 800 meters, we had about 10 times more oxygen. And at 1140 meters, we had 10 times again more oxygen. So each of these depths represents different oxygen. And we asked about the question of how does oxygen affect colonization and recovery. So what we found is no colonization at 500 meters, and this was not really surprising because the background community had no animals, macrofaunal animals at 500 meters. At 800 meters, where's my cursor, with oxygen just 10 times higher but still extremely low, we had only a few animals colonizing, and in fact these represent 4.3% of the background densities. Surprisingly, these colonizers actually came from quite a few different worm families, so there was some diversity but very low numbers. And then at 1100 meters, we had very high colonization, equivalent to 150% of the background community. But these belonged mainly to one species, an opportunistic worm called Capitella. This is a group that's known as a pollution indicator in shallow water. So it looks like oxygen affects the ability of animals to colonize, with the most colonization occurring at the highest oxygen levels. Okay. Now, we were also interested in how oxygen might control function, and we looked at the function of carbon processing in our colonization trays by introducing an isotopically labeled diatom, basically phytoplankton, which was dead, which represents phytodetritus, or equivalent to the primary production that might sink down to the seafloor. So half of the colonization trays deployed had this labeled phytoplankton, and what we found is that the animals, and we are able to track this isotopic stable isotopic label into the animals by looking at their delta N and delta C13 signatures. What we found is that only at the highest oxygen levels did we see a strong isotopic signal from our labeled phytoplankton. In other words, processing of carbon by the animals occurred mainly at this 1,142-meter site. The few animals we had from the 800-meter colonization trays had signatures equivalent to the unlabeled trays. In other words, they look like the background or the unlabeled. So what this tells us is that the crustaceans and worms that came into the trays at higher oxygen did more of the carbon processing, and not much happened at 800. And of course, we had no animals at 500 meters. But what we did sample in those 500-meter trays were some foraminifera. These are single-celled animals, protozoans, that took up large amounts of label. Let's see, here's the cursor. So here are the isotopic signatures of the foraminifera in our trays, and we see much higher values of delta N15 and delta C13 than we find in the, let's see. In the background community, which is the background values are here. And so what this tells us is that protozoans can take up carbon at very extremely low oxygen levels, but there aren't any animals around to do it. And then at 800 meters, we have protozoans taking up carbon and animals not. And at 1,100 meters, we have then animals kicking in. So along this oxygen gradient, we see a shift in who's doing the carbon processing. And these results are consistent with past research done by Woods et al. That shows that very low oxygen inhibits the carbon processing by animals, but that promotes it among protozoans, the foraminifera. Okay, so to leave you sort of with some bigger, to put this in context and leave you with a bigger perspective, we know that many of the world's oxygen minimum zones occur in remote areas that are unexplored. And so we know almost nothing about the dynamics of the benthic or seafloor communities. We do know from what you've heard already from the earlier speakers that oxygen minimum zones play key roles in ocean biogeochemical cycling, and they're also turning out to be very important repositories of microbial and animal diversity. And our results suggest that oxygen and very low oxygen can inhibit the recovery potential of communities and also and can affect the ecosystem functions. And so I think the final message is that as we increasingly begin to exploit resources in these environments, oxygen minimum zone, continental margins, we have to really proceed with caution and learn more about the ecology of the systems. And I'll stop there. Thank you.
Right, thank you very much. Are there any questions? No? All very clear? All right. I just wanted to tell you that the session about oxygen depletion will be tomorrow between 1:30 and 5:30 in room 24. And there will also be another interesting lecture over there about how the ear stones of fish, those are in the balance organ of fish, how they can help with this research. So maybe that's also interesting for journalists. Right, I would like to thank you a lot and good luck with the further research.
Thank you.
Thank you.